Now we'll start. Okay.
Yes.
Okay, let's start this session this morning, the first session. In this session, we have 4 speakers: Professor Disalvo, Professor Wen, Professor Zhang, and Professor Tanaka. Now let's begin the first lecture from Disalvo. He got his doctorate degree in biophysical chemistry in 1974 from National University of La Plata, Argentina. Now he's working as a superior researcher in the National Research Council, Argentina, and as the head of the applied biophysics area of the Center of Applied Biophysical and Food Research. His research interest is to analyze the structural and dynamic properties of lipid interfaces related to the water distribution in specific sites and its relevance to membrane stability and permeability of the cell in osmotic and regulation of the enzyme activity by experimentals and the molecular simulation. The title of his report is The Role of Water Activity in Thermodynamic Response of Lipid Interfaces. Okay, now let's welcome Professor Disalvo. Please.
Okay. Okay. Can you hear me?
Yes.
Yes.
Okay.
Yes, yes.
Thank you for the presentation. And first of all, I should like to can you see this first slide?
Yes.
Okay. Well, first of all, I want to thank the organizers and especially to Romiana for the invitation to participate in this very interesting meeting. And also to give my regards and my congratulations to Romiana on behalf of all the colleagues in Argentina that had the pleasure to know you when you visit us in 2015 in the context of a water symposium also. So I am glad that you are starting a new life, but I cannot see you sitting without doing anything.
Yes.
So that's what happened to me also.
Well,
anyway,
I learned from you, so you to blame. Thank you. Thank you.
Okay. Well, the topic of my talk is about the role of water activity on the thermodynamic response of lipid interfaces. And this work has been carried out in the Institute of Applied Biophysics and Food Research of the University of Santiago Estero and CONICET in Argentina. It is not necessary to know that sorry that I cannot move the slides. Yeah. Water is a unique molecule because due to the hydrogen bonds, the type of hydrogen bonds and the type of interactions, it has a high surface tension, a high dielectric permittivity, and a high heat capacity. That makes that as ideal solvent for the stabilization of biostructures such as protein and DNA and biomembranes. And this, what is also of very practical interest is that it stabilizes biomimetic systems of biomembranes that are usually studied at full in terms of lipid properties. In this regard, what the properties of lipids that we are interested in is about how they hydrate and how this water that hydrates the lipids have structural and thermodynamic consequences for from the biological point of view. Water in membranes has been found to be stabilized around the polar head groups, specifically the choline groups, the phosphate groups, and also along the carbonyl groups, as you can see in this blue line that show how water can penetrate into the inner bilayer. A detail of this organization is given schematically in this picture, in which we can define in a length of around 10 to 20 angstrom a region in which the phosphates and the carbonyls are surrounded by water, and from this plane, from the plane of the phosphate to the bulk, we can find several residues belonging to the several types of lipids that membrane contains and protrudes into the water phase. And this is also a very important point to deal. The question, the open question that I have in this regard with this description of water in membranes is how water properties are maintained or altered in the presence of biosurfaces, specifically the biomembrane or the biomimetics membranes. In what way these are relevant for biological functions, and if monolayers and bilayer systems are equivalent for structural and thermodynamic studies in terms of water dynamics. To answer this question, what we are proposing is to treat the membrane as a bidimensional solution of hydrated polar head groups that is above the hydrocarbon region. And in this interfacial region, with pH, because it's a phase between two phases, we can found the carbonyl groups, phosphate groups, hydrated, and they are immersed in a kind of water that we specifically name as second hydration shell. The idea is to treat this as a kind of ionic solution, but with specific features, because these groups are attached to the hydrocarbon change, and also are distributed in a plane. So the first step in this research was to try to have structural features of the bidimensional solution by means of FTIR and AIC spectroscopy and fluorescence analysis with a fluorescent probe such as Laudam. In AIC, what we do is to put a liposome dispersion, that is lipids in water, in an assembly of an AIC crystal that is set in a water activity equipment, and therefore we can measure the spectrum of lipids, and specifically the bands of water from high to low hydration, controlling very specifically, very accurately, the water activity in this receptacle. In this, with this kind of device, what we can take a series of experiments in which we can, at constant temperature, that is also well controlled, the hydration degree of the lipids, and we can obtain from this peak the area in relation to water to hydrocarbon change, and to make a isotherm that can be fitted by a Darcy and Watt isotherm, in which we can distinguish that is a population for strong sorption of with sorption and multilamellar water, that is multi-layers water in lipids. Also in the spectra, we can distinguish what happened with the phosphate groups and the carbonyl groups and the CH2 groups when they are hydrated or dehydrated, depending on the experimental design. What we can say is that the hydration of PO groups varies with the phase state and with the acyl nature. That is, that the hydration of the phosphate group is not independent of the acyl chain region. In addition, we can see that the hydration of, along the hydration or dehydration, we have a variation also on in the vibration of the CH2 groups, denoting that when the hydration is very high, the CH2 groups are separated by water molecules, and when the hydration is very low, they interact with each other with no water in between, and then it's a nice way to show how the PO groups hydrates in parallel to the CH2 groups in terms of the same water activity. We can see that there is a long extension in which PO groups hydration does not change too much, but the CH2 is changing from the very beginning of the dehydration. The same happened at the phase state, liquid crystalline, but well, it reports the same information. What is very important is to see that the coupling of adsorption at the phosphate group and the CHU depends on the presence of the carbonyl groups. We can see that this dependence of the frequency of the CH2 and the phosphates runs very different when we have ether lipids like this, are DPPC lipid, that is when we have a carbonyl group. That means that in the presence of carbonyl, the relation, the coupling between the hydration of phosphates and acyl change are different than in its absence. It is a point that will be important to deal later. In another line of experiments, what we do is to measure the phase transition of the lipids, because at the below the phase transition, the gel below the phase transition, the gel state is very poorly hydrated, and above is very highly hydrated. And in this case, we can see that the vibration of the CH2 increases sharply for different kind of lipids when we plot them in terms of reduced temperature. And we can see that the CH2 groups increase the frequency, meaning that the CH2 are separated by water. In the same range of temperature and for the same lipids, we can measure the polarization of water. And in this case, what we are seeing specifically is how the water outside the hydration of the hydration shell of the phosphate and of the CH2 behaves in terms of the transition. And we can see that when we go from the gel poorly hydrated to the liquid crystalline highly hydrated phase, we can see that a relaxable population of water appears very sharply. And on the other way, that when molecules like cholesterol that are able to rigidize the membrane to increase the packing of the membrane, this relaxable population decreases a lot. Moreover, the same spectra allow us to calculate the phase transition taking into account how the water balance in the phospholipid mixtures evolves with temperature. That is, we don't see the phase transition at the lipids, but we see the phase transition at the water near the lipids. And we can follow it and see something very interesting, that we measure the temperature along pure water, there is no transition. And when we measure it in terms of lipids, for instance, the DMPC, we see a sharp transition that corresponds to the transition from highly organized hydrogen bonds to molecules with much less hydrogen bonding. And this temperature is exactly the same that the lipid has. That is, the lipid has a transition, and water reflects that transition. That is, water is a mirror of what is occurring in the lipid membrane, as many times Romiana has remarked. So these transitions are due to the difference reorganization of water in terms of how water are interacting with the different parts of the membrane. And we can see that it's a strong difference in when the membrane is in the liquid crystalline state, the gray bars, and when they are in the gel state, the black bars. This has been published also in a journal in which we also counted the number of hydrogen bonds that each molecule can do, and we have certain information that is beyond the purpose of this talk. So we can arise to a partial discussion, and we can say up to the moment, at least in this brief resume, is that there is an exchange between of hydration and water of hydration and confined water that explain the plasticizer effect of water in the lipid membranes. It is well known that when phosphates hydrate, they expand the surface, and then the hydrocarbon chain has more degrees of freedom. But the new things that we are exposing here, that this is a coupling that depends on the carbon. The hydration of the phosphates and acid chains is modulated by the presence of CO groups. And in the presence of CO groups, when we add cholesterol to the lipids, we have that the cholesterol block the CO effect. And also, the last part was that water acts as a mirror of the lipid phase transition. Now, we are in condition to explore a little bit about the thermodynamic features of bidimensional solution at the interface. For that, we have to change a little bit our ideas about the biomimetic systems. One of the biomimetic systems that is usually applied to study the lipid behavior and to relate it with its physiological properties is a monolayer in a Langmuir blotched monolayer balance. And we can compress and expand moving a barrier. And then we have the surface pressure as a result of the difference in the surface tension of pure water and the surface tension with the lipids. Most of the models in which we can interpret the behavior of this monolayer has been treating the monolayer as a gas spread on the surface of water. However, this model is not very accurate to reproduce the isotherms cones of compression, and that is, in our understanding, that is because water is disregarded, and there is no consideration of the interaction of water with the lipids. So in terms of incorporate water as one of the components in the membrane, we treat the membrane as or the monolayer as an open phase in which water can exchange due to the compression. When we compress move the barrier and we compress the monolayer, there is a water outflow, and when there is an influx of water, there is expansion of the monolayer, and therefore we can express this in terms of the Onsager coefficients, in which the flux of water is due to the chemical potential of water and the surface pressure, and the changes in area is also due to the changes in surface pressure and the chemical potential of water. And we arrive to this coefficient, to this equation, in which we can put the surface pressure in terms of water activity, and these coefficients give us the interaction of the diffusion of water in water, the diffusion of lipids in water, the diffusion of lipids in lipids, and the diffusion of water in lipids. There is a difference between this coefficient because it means that the lipid-water interaction is lipids surrounded by water, and this coefficient, the diffusion of water in lipids, is that water around the lipid. So with this equation, we can fit very well the experimental surface pressure area. This has been published in +1 some years ago, and we can see that with the coefficients that we have explained before by adjusting, and these coefficients are not adjustables, but is information about the diffusion coefficient of lipids in lipids and water and lipids in water. We can fit very nicely the isotherms, the experimental isotherms, both in the liquid expanded state and the liquid condensed state. Now, the another interesting point that brings about this equation is that we can test the equivalence of monolayers and bilayers, because in monolayers we can fix the surface pressure, and with this equation we can calculate the water activity, and in a bilayer, for instance, in a lipid vesicles, we can fix the water activity by fixing, for instance, an hypertonic or hypertonic stress, and deduce from there the surface pressure of that lipids in the bilayer. From polarization of measured with Laurent, with the experiments that I showed you before, we can see that the same polarization is formed for monolayers and bilayer when we have a surface pressure of 30 millinewton per meter. So at this pressure, we can say that monolayers and bilayer are equivalent. And there are another test of enzymology that corroborate this. So with this formalism, we can argue now what is the role of the water activity in the responsive of membrane. What this means? This means that we always, or it is usually considered that the membrane have a passive role. That is, it's just a barrier for permeability. However, a lot of phenomena can be explained because the matrix of the lipids changes along the interactions of what we call a bioeffector. For instance, the injection of amino acid or a crop protectant, or a peptide, or even an enzyme. So in this picture, we have taken an example of the injection of a bioeffector underneath the monolayer, and see what happened with the surface pressure? The surface pressure increases, meaning that the surface tension decreases. And and when we reach to the equilibrium of this effect, we can plot the surface pressure increase in terms of the initial surface pressure. And the initial surface pressure gives an idea of how packed are the lipids. And when I am talking about packing of the lipid, I am talking about how is water entering into the interface. At very low areas or very high pressures, we can found that it is water that can penetrate into the acyl chains, and when the water is completely exhausted by the increase in surface pressure, there is no response of the membrane, and because there is no water, loose water, the relaxable population that I show you with Laurdan, and that is that can make the membrane respond to that bioeffect. So again, if we take this equation, instead of plotting the surface pressure, that is the membrane response in terms of surface pressure, we can put it in terms of water activity. And these are the curves that we obtain with the same data, and these data are important to see because you can see there is a slope very different for the same lipid, the same acyl change, but with that differs when the carbonyl is absent. That is, again, the carbonyl is a group that can modulate the hydration of the interface and have its consequence in the response of the membrane. When this is an schematic pictures in which the membrane response is therefore related to the confined water, but this response is modulated for lipids that has carbonyl and lipids that don't have carbonyl. Again, the carbonyl group is very important to modulate the exchange of water between hydration and confined water. Now, if we study with a little bit more detail the kinetics of how this monolayer evolves from the injection up to the equilibrium, we can plot this kinetics in terms of the Fickian or non-Fickian process. I will explain a little bit what this means. We can see that in the case in which we have 30 millinewton per meter, we have a Fickian response. That means that the membrane is not altered by the diffusion of the bioeffector. However, when we release the surface pressure and the solid found in the membrane more water, we have a deviation from the Fickian behavior. And this relaxation process can be, sorry?
We have just 1 minute just to speak.
Okay, yeah, I finished. This is one of the last. And the same happens when we have cholesterol. With the increase of cholesterol makes the membrane has less water, and the behavior turns into a Fickian one. So the implications and the properties of this membrane in terms of open systems is that we can exchange water by in and out by osmolytes, by the dissolution of polar solutes in the interface, or with solutes by mimicking water, like the case of trialols, or to change the membrane composition may bring in terms of further studies of the bilayer, is that we can think in the interaction of at least one of the monolayer of the bilayer with a cytoskeleton or cytoplasms that is known, that is a gel state, and to see how this behaves in terms of asymmetry and contact with cytoplasms. Therefore, I want to thank to all the collaborators, especially to Dr. Frias and Sebastián Rosa, the FTIR experiments, Jimena that makes the monolayer experiments, Perez that made the surface, the fluorescence experiment. Thank you very much for your attention.
Thank you. Thank you for your interesting lecture. Now, the time is 1 minute later, but it may have a quick question or comment.
Okay.
Is there anyone have a quick question?
Can I ask a question? Hello.
Hello.
I'm here.
I have a quick question. Have you ever examined the effect of ion composition on the interaction between lipid and water? For example, to compare the sodium-rich solution and potassium-rich solutions.
I have a very bad connection now, but I understand that you are asking about the effect of ions.
Yes.
Okay. It depends of the type of ions. For instance, the more important ion that interacts very strongly with the lipid bilayer is calcium.
Mm-hmm. Yeah.
In the lower extent is magnesium, and
how about sodium or potassium?
Ions, sodium and potassium has no specific. Sodium and potassium has no effect, at least as far as we know, what we have found in the hydration sphere of the phosphates. It's not able to change the hydration, not in the carbonate. It has a screen effect on, for instance, the charges. I don't know if
I have a comment. As you pointed out, there is no direct interaction between sodium and lipids, but you know, there is an interaction between sodium and water.
Yes.
So indirectly, as a result, lipid dynamics is changed.
Yeah. Well, that is a very interesting question, and it's a very old question also, because sodium is considered that is water disruptor, and potassium is a water structure making ion. But at the level of the interactions of the acyl change, at least the effects are not so clear in this, at least with these tools.
Okay, thank you very much.
Okay. We may have no time to speak more, Professor Santiago. Anything?
Yeah, just one comment that we had lectures. Thank you very much for the talk. It's always fascinating. We had yesterday a lecture on biomaterials and biocompatibility. People are talking about the intermediate water. And I saw your slides with hydrating water, intermediate water, and bulk water. And it's exactly the image that we have about distribution of these water species. So I'm very happy to see this. And maybe this is a direction that we have to relate our WOMACs and water species that we discovered, describing each of those bands a specific functionality in the future.
Yeah.
Thank you. Thank you.
Yeah.
Marita,
Okay.
thank you also.
We have no time to discuss more. So
Yeah.
now let's move to the next speaker. Thank you, Professor Disalvo. The next speaker
Thank you.
is Professor Wen.
Thank you.
Thank you very much.
Okay.
Hello, good morning.
Good morning.
So let's introduce your situation. Dr. Wen achieved his PhD in food chemistry and technology in 1999 at the National Source University. I cannot read the original text you provided because it's in the language of Argentina. The university, the National Source University, is right.
Yeah.
Since his postdoctoral stage at the Institute of Biochemistry, he's working now in biophysics of membranes, dealing with the architecture of the membranes, their physics behavior, lateral organization, and